And so before starting my talk, I'd like to say thank you very much to the specialist Murase-san for inviting me to hear their exciting interdisciplinary meeting. So it's a very great, my great pleasure to show you all our research work. So my research field is structural biology. The structural biology is a field studying the biological functions based on the biological macromolecular structures in the atomic level. So you know that in cell, many of these biological macromolecules, such as proteins and the RNAs, are working and do these many biological processes. All those biological processes are also depending on the three-dimensional structures of all those molecules. So these molecules sometimes form a very huge complex. And so we call those complex super molecule or the macromolecule nanomachine or biological nanomachine. Also here, we showed several examples of those molecular nanomachines. So they are in polymerase and the ribozyme. So you know that this year's Nobel Prize of the chemistry field was awarded to the structural analysis of this ribosome. And so the virus itself is the huge macromolecular assembly, the protein assembly, and that is, so you know that it affects the cells. And this is my research target. It is the bacterial flagellum. It is a huge molecular nanomachine. Okay, so here I show the swimming of the bacteria. Bacteria swims very actively in the liquid environment. So you can see some tails, although the spots show the bacteria, and you can see some tail. So this is the bacterial flagellum. So it's a bundle of the small filament. It's a dark field microscopic image, so you can see the very thin things are visualized. Okay, so this is the electron micrograph of the bacteria. This is the salmonella. Also, the salmonella has several filamentous things. We call this the flagellum, and this is, you can see the wavy form. So in case of salmonella, so these filaments form a single bundle. And at the base of this, each filament, so we have a molecular model. Here is a magnified image of the detached bacterial flagellum. So you can see all some rings and the filaments. And so these ring parts form a rotary motor. And so each motor rotates, and so then the boundary is formed, and so making a big thrust power to propel the bacterial cell. Okay, so what is the size of this stuff? So let's think about the size. If the size of the bacteria is comparable to that of the human, so let's say 160 centimeter tall is my tall. So then the size of the diameter of each filament is about 2 centimeters. And then the bacteria swim in the liquid environment. So in this size, so the water molecule, the bacteria feel the water molecule is not the sort of the continuous liquid, but it probably feel the sort of the particle. So then probably the bacteria feel that the water molecule has like a sand particle with 0.3 millimeter diameter. So then the bacteria swim in a very viscous environment. And the swimming speed. The swimming speed is 20 micrometer per second. So it's corresponding if the size of the bacteria is the human torso, then the swimming speed is about 60 km/h. the 60 km/h, and then so the bacteria suddenly stop and change to its swimming direction. So because it's living in the really highly viscous condition, so in this size, well, the inertia of the cell, the contribution of inertia is very low. So then the cell is very smooth swing, and then so suddenly stop, and then swing the other direction. Okay. So the bacterial flagellum is the first example of this rotational mechanism involved in the biological system. So this is this kind of experiment to prove the bacterial flagellum actual rotation. So you know the other, so eukaryotic the flagellum, such as sperm flagellum, that is very different from this one. So there's such kind of flagella, so making us the actual flagellation, not the rotational motion, but the bacterial flagellum actually rotate. So the filament is attached on this slide glass, and then so you can observe this kind of rotation. And then so you can see that the rotation direction is sometimes to this the backward and this the forward rotation. So this motor can switch the rotation direction. So what is the source of the rotation energy? So the bacterial flagellum so rotated utilizing the proton, so-called proton motive force. So using electrochemical potential difference between the outside of the cell and the inside of the cell. So that the protons, so from also the cells pumping out to the protons, so from the inside cell to the outside, so using the respiratory complex system. So then the pump out to the protons. And then so you have the electrochemical potential so between the outside and the inside. And then so the influx of the proton so can do many works. So for the flagellum rotation, and in other case, so the proton influx utilized for the synthesis of the ATP, the very important small compound for the living organisms. And so this machinery is widely distributed in the living cells, and therefore so it's inverted FATPs. So this enzyme also appeared in my talk later, so being my mapping. Okay, so this is the schematic drawing on the motor part. Well, from the electron microscopic observation, each part, rough structure of each part were known. So in the bacterial surface, so there is in the membrane, and so they have the peptidoglycan layer that supports the strength in the given strength of the bacterial cell wall, and so here is the outer membrane. So then so there are so many ring structures, and so this part is the bushing, a sort of the bearing to the driving shaft. And so here, so we have some rotation of rotor part of the motor, and here, so we have the stator, and the proton, the flux goes through this way, and the torque, the rotational torque is generated between the stator and the rotor. But so the details, the mechanism are not well known. So because the atomic structure of these parts is still unknown. Well, the rotation speed of the flagellum is about 300 Hz. That means that the 18,000 RPM. So just imagine the automobile, the car's engine. So it's corresponding to almost the F1 cars. So it's really quick. And the other the flagellum motors, such as the marine bacteria, marine vibrios, so that utilize actually the sodium ion influx. So it rotates really quickly to the 1500 hertz, so it's very quick. So the rotational mechanism is which we seek to analyze, but the structure is not known, so the details is not known. Well, so here I show the schematic drawing, the whole bacterial flagellum. So here I show the previously the motor part, and this is the filament part. Well, the bacterial flagellum is a huge molecular assembly that is composed of more than 20,000 protein subunits. And it's actually about 13 different kind of proteins are made up of this amazing structure. And so each part has its own function. The filament so acts as the head of screw. And here we have a universal joint. So the rotational torque is generated by the motor part, and that is transmitted by the drive shaft. And then so this rotational energy is effectively transmitted to the screw. But so the rotational direction and also the direction of the filament and the axis of the motor is not so linear. So in sometimes the change in the direction. So to transmit the rotational torque to this filament, we need a universal joint. So there is like this. So then so this all the parts has an amazing mechanism. And so this mechanism is based on this, its structure of the component proteins. Okay. So then the most well-known part of the bacterial flagellum is actually this filamentous part. So in my following talk, I'd like to focus on the structure and the function of this filamentous part. And so latter half, I'd like to focus on this part, so-called export apparatus. That's actually found that some evolutionary relation to the other macromolecular machines. So as I showed you that the bacterial flagellum, it looks so very unique machine. So it seems no relation to the other biological system. But recently, we found the structural evidence that the bacterial flagellum is evolved from the other macromolecular machine. Okay. So the latter part, I'd like to focus on this evidence. So then the normal part, well, the bacterial flagellum filament. Okay, so as I showed you that the filament formed this kind of bundle and thrust the water and propel the cell. Okay, so again, so we see the swimming motion of the bacteria. So you can see that so here, so the bacteria swim and then so quickly turn to the other direction. At this when so it turns in the direction, you can see this these filament bundles apart. So then something occurs this stage to change the swimming direction. So what's happening? So bacteria swims straight forward, so in bundle form, and then so when it changes the direction, the filament apart. So at this stage, so the rotational direction of each motor so reverse. In the rotational direction in the spinning mode is the so-called counterclockwise motion. And the helical wave is a left-handed pellets. But So when it changes its direction, the rotational direction changes to the clockwise. And at the same time, this wavy form, the helical form, is switched to the wide-handed helix. So because if the helical form is not switched, so then if the helical form is in the left-handed manner, so then just invert the rotational direction. Then the wave of the helix toward the south. So then it causes a sort of jump at the base of the filament. So then the filament switches to its direction. Okay, so to thrust the water into this waving form and this rotation, and you can thrust. But so if you change this on the rotation direction, so then this wave comes to the south. So then it jumps over. So then what the cell do is this helical form, superhelical form, changes this way. So they're scattering apart. So the bacteria do these things like this. Well, so this property is actually the intrinsic property of the filament itself. So this is the detached multifragile filament. And so we have some viscous form. In response to the viscosity, the filament changes its wavy form. So here, so we have a short pitch and a long pitch, and suddenly changes it at the short pitch. And this supercoiling form is changed in response to the other environmental conditions, such as the ionic strength, the concentration of the salt, and pH. So here, we can see the wavy form is changed to the straight form. Why so this kind of structure change occurs? So actually, the bacterial fragile filament is composed of just a single protein, flagellum. And so the filament is the helical assembly of this single protein. Just imagine, so the helical assembly of the single protein, just to make a straight tube, do not make the wavy form. But so if you assume that the two-state, so you can make a wavy form. Also, it's a helical assembly, but if you change it as your viewpoint, so the bacterial fragile structure can be described as a sort of assembly of these protofilaments. So the flagellum has 11 protofilaments, so they assemble into the filament. And if you assume that two different kinds of form, so one having longer repeat, and one is short. So you have a curvature. So just imagine a straight protofilament, and the one is the short repeat. So you have the donut shape. Okay. So if you have tilt of this protofilament, one is left-handed, the other is right-handed. So if you mix, so that you have no more as a donut, but this pod shape. Okay. So the mixture of these two types of conditions produce a superhelical form. Okay. So if you change the number of these compounds, so you have the various supercoiling form. Okay. So this kind of model is proposed about 40 years ago. So it seems so reflected in our actual situation, but no evidence. So according to these theories, you must have two different types of straight filament. So then there exists. So in nature, so we isolated many different types of single mutants in the flagellum. So that forms a very different type of supercoiling form. And some are formed just a straight filament mutant. And so those straight filaments can be divided into two groups, L and R. And so then the helical packing parameters of these two types of filaments were analyzed using the X-ray fiber diffraction method. Then so what found is the difference now the repeat distance difference between the longer repeat and the shorter repeat is only 0.8 angstrom. And so we can exactly measure how it is the tilt angle of this outer and the outer. And then so we should simulate the superhelical formation, assuming that these two types of conformation. So then so here the result. Well, the flagella is composed of the 11 plot filaments. So then so the 12 different supercutting forms is expected, and here shown those supercutting forms, and we compare the natural supercutting form of the peaches and also the twist and couch, and so here, so we have the theoretical point in the white circle, and observe the helical parameters plotted on this figure, and so the both are nicely fitted. So then so this is experimental evidence that is supporting the original idea, explaining to the why such kind of super helical form is produced. So then so this type of switching, so actually in response to the changing of the rotation direction. So what type of force is compared to the making the conversion of these two conversion change? Well, the quick reversal of the rotation, so making the twist first. And then so probably these twist force converted to the sliding force between the neighboring filaments. And this may be caused some, turn on the switching of the convection change. And that is propagated along the filament. So then the dynamical change occurs. So then what is the kind of switch? So to analyze this, so we need a more detailed structure of the bacterial flagellum. So to analyze the atomic level structure, we must crystallize those proteins. But so the flagellum forms a filament, and so the normal crystallization condition, so it just tends to form just a filament. So then, we overcome this problem. The hint came from the actually low resolution electron microscopic analysis. Well, if you chop off the N and C terminal region of the flagellum, so then you have the filament, but some have a distorted filament, and so those filaments missing the inner ring structure. Also, actually, the bacterial flagellum has a double tubular structure with a protruded very outer part. But the inner core part is composed of this the double tube. And just the inner tube structure is destroyed. So if you chop off the N and C terminal regions. And but still have so it depends on how much you chop off those terminal regions. But if the small amount of terminal region is cut. So then you can still have the filament and the inner tube structure is disappear. But so if you use these kind of modified proteins, just mix with some normal filament, it can make this wavy form. That means that to make this wavy form, so I mean this on the left and the right, L and R type to conformation. So the important point is this outer ring region. So that is the N and C terminal region is not required to make determine the L and R type conformation. So then, we chop off it's N and C terminal region, a little larger. So that fragment cannot form the filament, and then we succeed to crystallize it. So then, we got structures like this, and fortunately, we have the protofilament in the crystal. So what's happened? So here, it's shown. So our crystal is a part of the protofilament. So up and down manner, and this up and down conformation forms some sort of sheet, and this sheet started to form a 3D crystal. And so if these filaments aligned with the same orientation, so you have native filament. So then we get a protofilament structure directly from the crystal structure. So then we fit this protofilament into the low resolution electron microscopic image, and it's nicely fitted. And the repeat distance of this protofilament was 51.9 angstrom. This is exactly the same as the R-type, shorter type conformation protofilament. But we cannot make the crystal that has some longer type protofilament. Probably the longer time protofilament is not so stable, and the most stable one is the R type. So probably in the filament, this will sort of switch to the shorter repeat to the longer repeat. So then, after that, we are now planning and planning to making some mutant to inhibit this kind of structure switch. It's now undergoing. And after that, in 2004, the technology using the electron microscope is dramatically improved. And we had whole filament structure of the bacterial flagellum, using the electron microscopy. Well, the resolution is not the actual atomic level, but you can trace the main chain of the amino acid of this protein. So we can construct the whole filament structure. So in the crystal structure, we are missing this inner part, but this structure gives the information, as well as the inner tube part. So then, using this model, actually the structure is just solved the shorter repeat type one. But they constructed the whole filament model. So then, the original molecular dynamics simulation was done. Using a short segment of the filament model, and also this is a collaboration with Professor Kitao in Tokyo University of Tokyo. And what he did is also we have just a straight filament model of the archive, and for twisting force, load and twisting force in the both end. Distort to this segment model in silico to the starting from this to this various point now with the helical form. So then, so suddenly release the such kind of form in silicon, and then so do this the molecular dynamics simulation. So that means the relaxation. And so then, so where this segment structure is converged. So then, so what he found, it converged this certain region. So one is around here. This is exactly very near to the predicted region that reflect the normal swimming mode supercoiling form. And others is so near to the so let's say in a semiconductor power. So this is some tumbling time. So the filament take this form. So so this means that the atomic structure so directly so explain and related to the actual so native biological structure change. And ultimately understanding for the molecular mechanism, so based on the structures, we must solve the actual longer type structure, cell type structure. This is almost finished with the structure work. And after that, we mix and model the actual curved superhelical structure in silicon, and get to the information from those simulations, and so back into the biological experiment, such as the nutritional experiment, to full understanding of the so this mechanism. But it's really ongoing. Okay, I spent too much time for the filament part, the latter parts of the micro. Well, so the filament, the flagellum is so has an amazing structure. And so that seems attract to the creationists, not to the sort of Darwinian evolutionists. Well, so if you type the flagellum evolution as a keyword and so just do the Google search, you can find many different type of the home pages that is produced by the creationists. Well, so I remember seeing this picture. But I don't have any friend of the creationists. But you think that our movie is over there. So what's happening? So I'm Japanese and I learned that actually really happening. So. Well, so that is an ID movement. So probably the US people so well know that this movement. Well, the creationists starting this movement that's in the 1990s. So then this is the intelligent design. So such as the incredible the structure, the amazing structure. So it's too complex. So there must be some designer. So then the creationists strongly claim this part, and so they say that the incredible, irreducible complexity. So bacterial flagellum is so such as a complex structure, and if we so there's some parts, lack of some parts, it should be lost to their function, but it's not true. So in some mutants and some different type of organisms has a really different type of flagellum that is lacking in the certain parts. And the other claim, the apparent absence of the non-fragile homogeneous fragile component provide evidence of ID. It's also not true. So what I'd like to show you later is the relationship between the other molecular machine and bacterial flagellum. So I must very rush and quickly. So that is the spot apparatus. Also, actually, so well, the 1990L and 2008, well, they are found that is a very interesting structure. So that is called the type 3 needle complex. So the structure is very similar. So So here is the surface, we have the flagellum, but so sometimes we can see the flagellum like something. The people thought that this is the actually the flagellum under construction, but it's not. It was not true. That was proved by the Kubori-san, Dr. Kubori and Garand, and also what are they? So this is the so-called injection. What is injection? So the virulence bacteria so utilize this machine to infect their host cell, and they produce synthesize the virulence factors in the cell, and so these proteins are directly so injected into their host cell using this molecular machine. And the shape and so the corresponding proteins is very similar to the bacterial flagellum. So here the name of the protein, and here so show the component protein of the needle complex. Here so show the component protein in the bacterial flagellum. There are some duration, a very deep duration. Okay, so the export apparatus in case of flagella is composed of these actually 9 different proteins, and the stoichiometer is not well known. And so it can be divided into the 2 categories. One is the membrane component proteins, and the other is the cytoplasmic component proteins. And these cytoplasmic component proteins dynamically so change its conformation or its binding partner to transport the substrate to the outside. I forgot to tell you that bacterial flagellum is the supermolecular structure that extending from the cell. So each component protein is synthesized in the cell, and it must be exported from the inside the cell to the outside. So then the bacterial flagellum has their exporting machinery. And the component protein of these are very similar to this needle complex. And so in this component protein, we have some ATPase. So then we try to analyze the structure of this ATPase. Well, this is the domain architecture of the ATPase, the name of the fly eye. In the middle part of this proteins, it shows this similarity to the F4, F1 ATPase alpha beta subunit. Okay, so let's take a look, a short look at what is F4, F1 ATPase. So I showed you the previously that the F4, F1 ATPase produce synthesized ATP, so utilizing the proton influx. And so here we have alpha and beta subunit. It forms the hexamer. So so here we have the gamma subunit that is penetrating this hexameric ring structure. And so the amazing synthesizing reaction occurs in this molecular machine. The proton influx comes, and this produces a rotation of this part, and this is the gamma subunit is rotated. And this rotation, so somehow the coordinated conformation change, coordinated conformation changes to the this ring alpha beta structure. So here, so we have this binding site of this ATP or ADP. Okay, so this is actually has no how to say hexameric or trigonal trimeric symmetry, but very distorted. So one side is empty, and one is occupied by ADP, and the other is occupied by ATP. So then the rotation of the gamma subunits causes a systematic conformational change to these subunits. So then the ATP is synthesized during this motion. Empty part trap the ADP and inorganic phosphate, and then so the synthesized ATP occurs, and then so after the final compound is released. So So that is controlled by the rotation. Okay. So we solve the structure of our fragile entities. Well, this part is very similar. So this is expected from the sequence, but the entire structure is almost the same. So that is amazing. So if there is such similarity, so implies that what the molecular mechanism may be the same between the FINTPS and our fragile system. So if so, then we must find the gamma summit. And very recently we found. So this is another component protein, flagellum. So here it is the central stop in the FOFINTP gamma summit. You can see the very similar structure, the central part. And after solving this structure, so we compare the both sequence. Just as the normal sequence alignment, we cannot find any similarity between them. But after finding this 3D structure, we found the very locally conserved region that is around here. So then after that, so we observed the electron microscopy with the mixed Fly and Fly J, and we found that finally that the Fly J is located in the central part here. So then, well, so now, so we know we found that this ATPS and the Fly J form almost the same structure as FINTPS. And actually, so there are some similar proteins formed between this system and this system. And so these proteins colored by purple has a strong sequence similarity to this colored purple protein. And this must utilize the proton flow or rotation. And very recently, so my colleague, Dr. Minamino, found that well, to export to the substrate protein, so actually the proton flow in flux is essential. So then, so we're thinking that this FOF1 machine, it is a very basic enzyme for the living organisms. That is, how this structure is located in the center of the larger. And we don't know how this motion is causing a protein export. But so the structure so strongly shows that so probably so that this amazing molecular machine, flagellum, partly evolved from this very basic, looks different molecular machine. So I'd like to close my talk. Sorry for taking a very long time. Finally, I'd like to show my colleague, and almost all these work is supervised by the cage number, the professor. It's actually my boss, cage number. And I'd like to thank you very much for your attention.